First of all, I want to thank the organizing committee for this invitation. The subject of my today's presentation is the role of GVAS studies in nephrology, and particularly in the reconstruction of the concept of chronic kidney disease. In recent years, we are faced with an epidemic of chronic kidney disease, the so-called CKD storm. Up to 850 million patients suffer from some form of chronic nephropathy. The prevalence of CKD is estimated to be 10 to 30% of the general population, and the major risk factors are diabetes, hypertension, cardiovascular disease, and obesity. These risk factors determine the two main forms of chronic nephropathy, diabetic kidney disease and hypertensive kidney disease, that together account for up to 50% of all end-stage renal disease cases. Of course, this is the classical view of chronic kidney disease, in which a major risk factor determines the diagnosis of the corresponding type of chronic kidney disease. Nevertheless, there are many clinical and epidemiological findings that put into question the existence of such diagnosis at all. Let's see the case of hypertensive kidney disease. Comparing the two main RRT renal replacement therapy registries in Europe and USA, we see that there is a great discrepancy between the incidence of hypertensive kidney disease as cause of end-stage renal disease between these regions. This discrepancy is also obvious in these European registries, where we see a great difference between the incidence of hypertensive kidney disease between the various European countries, and also between regions of the same European country, Spain. And what do we know about this clinical entity so far? We know that most hypertensive patients do not develop kidney disease, that countries with a higher percentage of hypertensive patients has a lower impact of hypertensive nephropathy on RRT registries. We know that there is a lack of clear benefit of intensive systolic blood pressure lowering on incidence and progression of chronic kidney disease. And we know that there is an important familial aggregation of CKD that is independent of major risk factors such as diabetes and hypertension. The second type of kidney disease that raises the same questions is CKD of unknown etiology. Comparing again the two main registries in Europe and the USA, we see the same discrepancy between the incidence of CKD of unknown etiology between these regions. And in this case, this great discrepancy is indicative of an arbitrary diagnosis and the possible existence of another causal factor. Indeed, in this memorable study of Emily Groupman and her colleagues, in which 3,000 patients with chronic kidney disease were studied, researchers found a genetic diagnosis in 10% of patients. They also found a genetic cause of a chronic kidney disease in 17% of patients with the initial diagnosis of a chronic kidney disease of unknown etiology, as we can see also in this image. So there seems to be an elephant in the room of CKD, genes. Now we know with great certainty that the genetic architecture of chronic kidney disease consists of a wide spectrum of multiple genetic loci that are associated with various kidney phenotypes. On one end of this spectrum, there are very rare and very highly penetrant genetic variants in a single gene that cause Mendelian or monogenic nephropathies. And on the other end of this spectrum, there are very common variants in multiple genetic loci that are associated with the so-called genetic predisposition of chronic kidney disease. This is the two categories of genetically influenced nephropathies, monogenic and polygenic nephropathy. Monogenic nephropathies are caused by up to 600 genes. They are responsible for 10 to 15% of CKD and end-stage renal disease cases, and these genes are captured with next-generation sequencing methods. Instead, polygenic, up to 300 genetic loci are associated with polygenic nephropathy and the genetic susceptibility to kidney disease, and GWAS studies, genome-wide association studies, are the main research tool in order to identify these genetic loci. And what are these genome-wide association studies? They are mapping methods that identify genetic variants associated with an outcome across the genome in an unbiased manner. They do not focus on specific genes but screen entire genome. They try to find a statistical association between a genotype, typically an SNP, a single nucleotide polymorphism, and an outcome, typically a kidney marker or disease. And the finding of a SNP in a patient group that shows a significantly different frequency from that in the control group identifies a candidate genetic locus that is likely associated with a kidney phenotype. This is one of the first GWAS study in nephrology field, where the researchers found a significant association between CKD markers and genetic loci such as UMOD, and SHROOM3. UMOD is the gene of uromodulin, a protein of kidney tubules. In this study, researchers found this SNP to be associated with CKD markers, and specifically, the minor T allele of this SNP was found to be associated with 20% reduced risk of CKD. In this second study of the same researcher group, researchers found another SNP to be associated with CKD markers, and specifically the minor C allele of this SNP was found to be associated with lower uromodulin levels, higher GFR, and reduced risk of CKD. So researchers has begun to translate this genetic association in a functional mechanism from gene to gene product. The obvious linkage between the gene and the gene product was investigated by Trudeau et al., who found that carriers of the UMOD risk haplotype showed twofold increase in UMOD transcript in kidney samples and increased levels of uromodulin in urine. In order to model this effect in vivo, they generated transgenic animals which showed increased levels of uromodulin in urine. And what they found? They found that increased levels of uromodulin expression cause hypertension, increased salt sensitivity, and increased expression of kidney tubule damage markers. And how is this happened? This has happened through increased activation of NKCC2 at TAL level that is caused by increased phosphorylation of NKCC2 and not by increased gene expression of NKCC2. So we have a functional model here. SNPs at promoter region of UMOD gene cause increased gene expression, increased levels of UMOD in urine that on one hand these increased levels cause increased sodium reabsorption at the TAL level and arterial hypertension, and at the other hand, they increase the vulnerability of tubulointerstitial space in kidney damage and chronic kidney disease through unknown yet mechanism. In the following years, other genetic studies were carried out who found significant associations between CKD markers and many genetic loci. This is a recent GWAS study that shows association of GFR based on creatinine with 424 lead variants, of which 348 loci were validated by association with at least one alternative biomarker and thus classified as likely relevant for kidney function. However, the big thing with these GWAS studies is not simply to identify genetic loci that are associated with kidney markers, but to translate these findings into biological mechanisms, clinical practices, and therapeutic targets. In this post-GVAS field, there are many methods that create a path from GVAS to biology and clinical practice. Statistical fine mapping methods, functional fine mapping methods, cellular models, all together permit to research partners, clinics, industry, and universities to walk with confidence on this path. To clarify this complex post-GVAS field, I have selected three crucial questions that need to be answered whenever a genetic locus is under the research microscope. Which are the causal variants? In which cell types do the variants act? And which genes are regulated by the variants? In this, this recent our review article at NTT Journal, we made an effort to summarize graphically the main GVAS genes that are associated with CKD markers or traits. As you can see in this first image, the majority of these genes are located at the tubulointerstitial space, and at this level, many of these genes govern biological pathways that end up in a common pathogenetic mechanism of CKD, tubulointerstitial fibrosis. This is an old knowledge of nephrology, but these studies have begun to reveal the genetics and biological components of this knowledge, and this will permit to the future to identify new therapeutic targets and to develop novel kidney treatments. So, what we have learned so far from these studies: genetic risk variants are not associated with the occurrence of major risk factors for CKD, such as diabetes and hypertension. Kidney damage appears to be mediated through a possible increase in the vulnerability of the renal parenchyma to the action of these nephrotoxic risk factors. Multiple small damage hits to the structural and functional integrity of kidney make the latter particularly vulnerable to the major stress from the action of major CKD risk factors, and the majority of the genes that are implicated in this vulnerability are expressed at the tubulointerstitial level of the kidney. This is the so-called tubulocentric view of chronic kidney disease. All this knowledge can be reflected in what I call Jenga theory of CKD. This is a normal kidney. This is a kidney with SNPs, with risk SNPs, and a genetic predisposition of a CKD. And this is a kidney with a genetic predisposition and or a dominant genetic variant. So we have two kidneys genetically compromised. When these kidneys are faced with a major risk factor, such as diabetes or hypertension, then kidney collapse, and we have the onset of chronic kidney disease. Thank you for your attention.